Hi, everyone. I'm Natalie Cohn, head of the OpenAI Forum community, and I'm so pleased you were able to join us this afternoon for another expert discussion on how AI is advancing science and laying the groundwork for solving some of humanity's hardest problems. What if we could predict disease before it starts, tailor treatments to each person, and discover new medicines in a fraction of the time? AI could unlock this future for human healthcare. Today, we're joined by three leaders helping make that future possible. Joy Zhao, Life Science Research Lead at OpenAI, will be facilitating our talk today, welcoming Grace Zhang, co founder and CEO of Perturb AI, and Xin Jin, co founder of Perturb AI, professor at Scripps Research. Perturb AI is working to transform biology into a predictive science by combining one of the largest in vivo brain datasets ever assembled with advanced AI systems that can help scientists explore more complex biological data in entirely new ways. OpenAI has been proud to collaborate with Perturb AI through our science efforts, reflecting how deeply we believe AI can advance scientific discovery along with world class research partners. That includes our work on GPT Rosalind, a new AI system built to advance life science research and speed biomedical discovery. Together, these efforts aim to lower barriers to discovery, expand who can participate in research, and accelerate the path from breakthrough science to new medicines and better care. Please join me in welcoming Joy, Grace, and Shin to the OpenAI Forum community stage.
So good to be in the studio with everyone. Thank you so much for the intro, Natalie. Grace and Shen, to start us off, what do you think is changing in biology right now that makes this moment feel different from even just a few years ago?
What I'm seeing is that biology is shifting from a descriptive science to a more predictive one, to a more predictive one. Quite different from math and physics that's full of theorems. Biology is a discipline that's with few organizing principles, but lots of exceptions. What I can recall a handful of theorems like the central dogma of biology, but whenever people ask me about biology, the question is always, it depends, right? Same mutation can have very different effects in different people. One gene can have a dramatically different functions depending on the cell types they're in. So for a long time, what people have end up having to do is to choose, what kind of questions they ask and be able to go really deep and measure those. But now with the technology advances, what I'm seeing is that we can finally start to approach the biology as a whole systems. And some of the technologies that I, were witnessing that allow us to measure biology much more realistically. For example, the sequencing technologies, the imaging technologies, finally allow us to see and see what things are and where things are. The single cell technologies allow us to measure things at single cell resolution instead of an average. And then the amazing work that has done by Human Cell Atlas and Allen Brain Institutes, giving us the opportunity to setting up the reference maps, what's happening in healthy cells and healthy tissues. Then, last but not least, the editing technologies allow us to make a change and see what happens. So we're at an explosive time of technology, and as a result, biology is changing for the better of it.
Very exciting. What do you mean by predictive biology?
Yeah, as we were talking about, biology is experiencing the transition from a descriptive science to a predictive one. So how I think about it is, when we talk about descriptive biology, is talking about what things are, but when we're talking about the predictive one, is what happens if something changes. Right? So, and for example, what happens if there's a genetic mutation? What happens if I take a medicine, and what's going to happen to my body? And fundamentally, medicine is a prediction and a perturbation problem. We want to see what happens if someone takes a drug. How is that going to affect their outcome?
Yeah, I think biology is dynamic. It's like having a map to San Francisco and New York City. These are sort of cartographies and generating a descriptive biology like Grace is talking about. But having the map doesn't mean you know how to enjoy New York City. So I think being able to generate these predictions to some degree, but really understanding the system as a dynamic entity is what's really where the therapeutics and very effective treatments might come from.
Yeah, and the ideally, if we have a way of measuring everything, then that would be great. But unfortunately, biology doesn't give us a single variable, right? It gives us so many variables in terms of the number of genes and number of cell types and tissues, and on top of that, the diseases. So the number of combinations is just astronomical. It's impossible for any one lab to measure experimentally what's going to happen if something changes. And this is where the predictive biology comes in, right? Without it, then medicine and biology becomes a trial and error problem.
Makes sense. Yeah, so biology has historically been very difficult to model and to predict. What do you think has changed now that makes prediction more realistic? Is it something that happened across data, compute, or AI?
Yeah, well, I think it's a combination of all three, if not more, right? We just talk about the data because of all the technology advancements. Then we can not only measure DNA, protein, what's happening inside the cell, but also longitudinally over time, over different spatial contexts and different disease contexts. So it gives us a systems view of what's happening in the biology. Then we talk about you mentioned compute. And for the first, nowadays, we can finally train models that we couldn't imagine a few years ago.
That's right.
And then lastly, AI, right, it's really just revolutionized our life. And Joy, maybe I want to talk about the GPT Rosalind, right?
Yeah. So Rosalind is really, I think, kind of the beginnings of a series of models that we want to release that is focused on helping uplift the life sciences and kind of accelerating research. I think the way to kind of like mentally understand what Rosalind is, still a reasoning model. So this is now kind of like a new biology foundation model. But the difference is, let's say you imagine that you have a maybe first year grad student trying to do a task in biology. So it can still access things like AlphaFold, or it can still do single cell RNA-seq analysis. But this first year grad student kind of still has a lot to learn, right? Even though it kind of has the foundations, there's kind of a lot of like tacit expert knowledge that is kind of the outcome of this process of getting a PhD. And so what we really want to instill into the Rosalind model series is kind of this expert tacit knowledge. And so I think ideally we want to see as the model is kind of beyond PhD level at all of these tasks. And it also just has kind of great intuition for, when to call these tools and just to have a really kind of foundational understanding of the biological and chemical world. But yeah, also I think obviously at biology, foundation models also still has a place in the world. And so Grace, I don't know if you want to talk a little bit about that as well.
Yeah, there's foundation models, predictive models, and a tremendous effort going into that from industry, academia, right? The work that's done at CCI, that ARC Institute, Zyra and Genentech, we've seen wonderful examples and lots of improvements. So I can only imagine, where we'll be just in the next year and the advancement in predictive biology. Yeah.
Great. All right. Well, thank you so much, Grace and Shin. With that, I'll let you present a few slides that you prepared, and then I'll jump back on in a few minutes to continue our conversation.
Great. All right. So we all know that 1 in every 5 adults will experience some form of serious mental illnesses in our lives, and that is over 60 million people in the country. And for the longest time, we know brain disorders, neurodegenerative diseases, and also other type of chronic disorders are very common and extremely costly. Many of them also are highly genetic. So these conditions impact a lot of people, and we still don't yet understand their genetic mechanisms well. So the question is, why is this so hard and so difficult? And after decades of beautiful human genetics research, now we know that there are a long list of genetic variants or genes and mutations are highly associated with these devastating diseases and disorders. The gene lists are getting longer as the gene discoveries are ongoing and the sequencing getting cheaper and faster and better. And yet, what we don't fully understand is how each of the individual genetic variants that actually manifest our disease risk or resilience by acting on many different cell types in our body. In other words, every single gene's job in different cell types in our body is just different. You can think about these different cell types like different citizens in a village. And their jobs can be highly specialized, such as the jobs of a nurse versus a teacher in a village. And when disease happen, they also have different jobs. Some serve as a first responder and some are compensatory. And these jobs sometimes can be really hard to replace. And in other words, we really need to understand how each individual mutation that confer disease risks act differently across different kinds of cell types. And putting together, this is really a challenging problem because we're asking is not just many of these disease risk genes, but also how individually impact manifests in different cell types. So what we're really interested in building is these kind of functional atlases. Grace just talked about the difference of sort of descriptive biology versus prediction biology. So just by looking at a static picture of the cell type or the villages, what we need to learn is more than that to build functional atlases in introducing CRISPR mutations. To ask when disease contacts kicked in, when disease mutation kicked in, how does different citizens in the village react or compensate and cope with the situation? This is obviously a dynamic situation that we really want to build this picture. But if we want to study many genes across many different cell types, you will realize that the number of the possible combination also become enormous quite quickly. And across tens of thousands of different genes in our genome, across hundreds, if not thousands of different cell types, this is at least a sort of 20 million unique combinations of the map to be able to discover how each gene acts differently across cell types. At the experimental side, there are great advancements in CRISPR technology and genomic phenotypic readouts allow us to actually build this space using technology such as Perturb-seq. But if we're doing these experiments in vitro by analyzing thousands or tens of thousands of genes across a couple cell type that we can raise on a petri dish, what we are really sampling is a very thin slice of this huge search space due to the lack of the cell type diversity that it's very hard to grow or mimic on a petri dish.
So at Perturb AI, we're building a new type of training data for biology and medicine. We use CRISPR to introduce perturbations directly in the brain and measure the outcomes at the single-cell resolution. So rather than just observing the association between genes and cells, we're now starting to understand the underlying logic. When you introduce a change, which cells will respond and how. The result is the largest in vivo CRISPR atlas of the brain. It involves hundreds of cell types, covering 2,000 risk genes associated with neurodegeneration, psychiatric, metabolic, and inflammatory diseases. Most of those genes have never been disrupted in the brain and definitely not studied at cell type resolution. And this scale dramatically opens up the new functional search space for predictive biology. Since we open source the data, the response from the community has just been overwhelming. Thousands of downloads from academia and industry, reflecting the huge demand for such causal de novo data. We've had lots of collaborations and then deep discussions with technology and pharma companies. We're truly gratified by the level of interest we're getting and very excited by what the entire community would discover. And our excitement comes from the fact that the level of resolution and then what this data reveals. What you're seeing is a whole living system of brain architecture interconnected with diverse cell types that can never be recapitulated in vitro, all captured at single cell resolution. You can even see the pyramidal neuron axons projecting all the way to the striatum in interconnected brain regions in their native context. And context is what matters here, because genes do not act in isolation, but function in cell types dependent manner, very dependent on the environment they're in, and then the biological cell states. And this is where we can start to learn the biology. Let me give you a concrete example of, let me give you the concrete example of the green 2A and 2B subunits of the NMDA complex. Before this data, most of knowledge about these two genes came from gene expression and human genetics. Green 2B is linked to intellectual disability, 2A linked to schizophrenia. But their disease mechanism remains mostly a black box. And our data helps shed light on this in this black box. What we found is that green 2B functions across cortical and subcortical neurons, whereas the green 2A function is much more limited, and this is completely consistent with the clinical phenotype. In addition, just because the 2A and 2B are part of the same complex, the functions are not interchangeable. What we found is that when they're disrupted, they have opposite phenotypes. And this underscores the point that those two genes need to be treated and drugged very differently. And this speaks to broadly the benefit of functional in vivo data. It provides a missing link between genetics to mechanisms and mechanisms to drug targets. And with this, drug discovery becomes much more informed and deliberate. We believe large-scale causal in vivo data will become the underlying infrastructure for system-level drug discovery and core training data for the next generation of AI models. This infrastructure is only as beneficial if we can make sense out of it. Within days of releasing this, generating the data, we were able to get critical insights. And this would have been very difficult with the traditional bioinformatics approaches, which are slow, tedious, and highly specialized. What I want to show you today is a different way of interacting with science. For this demo, I used our CRISPR allos as the input and analyzed it directly with ChatGPT and Codex. There are two parts to this demo. First, can AI help us understand the raw biological data quickly? And second, can it help us decide what matters? Let me start with the raw data. I uploaded the raw data directly into ChatGPT. For the demo, I used a subset and asked ChatGPT to summarize the data, assess the quality, and suggest the next analysis steps. Right away, it recognized this is not ordinary single-cell data. It's a CRISPR perturbation data set with both gene expression and CRISPR guides captured in the same cells. It correctly identified 8,000 cells, 19,000 genes, and roughly 2,000 target groups. So immediately you can tell this is a high-resolution causal data set. It also recognized the RNA quality looks strong. High complexity transcriptomes, lots of genes detected per cell, and very low mitochondrial signal. In other words, the dataset is clean enough to support real biological interpretation. Without me telling it anything, ChatGPT correctly inferred that these are mostly brain cells, primarily excitatory and inhibitory neurons, directly from the marker genes in the data. This is already a meaningful biological readout. From there, it suggests the next set of analysis that a good computational biologist would do. For example, that cell type matters, that you shouldn't do any analysis that treat all the cells the same way. The takeaway from the first step is, with one natural language prompt, I can quickly understand both the quality of the data and the biology inside it. Good data makes the biology readable, and AI makes it much faster to read. But processing is only half the story. The bigger challenge is turning the output into hypotheses that actually guide the next experiment. Traditionally, this means reading papers, cross-checking databases, and manually stitching together a story for each gene. It works, but it's slow and honestly pretty painful. This is where AI agents becomes very useful. Let me show that with Green2B. This report is generated by a small team of agents. One looks at the biology, which cell types respond, and what may be going on mechanistically. One looks at the clinical relevance, what has already been tried, and what the risks may be. And then a grader puts it together into a simple decision signal. In this case, the biology agent shows something important. Green2B is not affecting all the brains evenly. Its effects are concentrated in a specific set of neurons. This is much more useful than knowing something just happened in the brain. And this is only visible because the data has real cell type resolution. The report also shows what's changing in the cells. When Green2B is disrupted, these neurons lose genes involved in synaptic communication, basically genes that help neurons signal and connect. This fits what we know about Green2B biology, so the results make sense. Then it compares Green2B with Green2A. These genes are part of the same receptor complex, so you might think they would behave similarly, but they don't. Green2A looks more like a counter signal. This is exactly the kind of distinction that matters for drug discovery, because it tells you these genes are not interchangeable. The clinical agent makes the biology more practical. Green2B has already been tested in the clinic, and the broad attempts to block it have not worked well. So if our data looks like reduced Green2B activity, more inhibition may be the wrong direction. And this is the kind of insight you really want, not just what to pursue, but what not to pursue, before spending years going down the wrong path. Finally, the grader turns all of that into a simple decision to help us prioritize. And that matters even more at Alice scale. With 2,000 genes, the challenge is not finding one interesting story. It's deciding systematically where to focus. That is what this index page is for, turning thousands of the reports into something a scientist can navigate. We can filter, we can sort, and we can search. HTT is a famous Huntington's disease gene and reassuringly scores very high on clinical relevance. I'll stop there, although there's much more we could do. For example, running synthesis agents across all of the reports, generating visualizations, and so on. But I hope this gives you a glimpse of the future. AI agents and reasoning models working together with scientists to turn raw data into high-quality biological insight. The goal is not just for faster analysis, it's better science, getting to the biology faster, more systematically, and in a way that's much more accessible. And this is exactly what we're building at Perturba AI. Beyond a single data set, beyond a faster workflow, a new discovery engine, combining high throughput in vivo CRISPR with artificial intelligence to decode biological intelligence and build better medicines. Let's bring Joy back on.
All right, thank you so much for your presentation. I also just think it's really funny how you can tell when ChatGPT has written a HTML report because it always uses the same color scheme. Yeah, I was like, okay, this is definitely from ChatGPT. Let's connect this back to human health. How do you think that predictive biology can change what's possible in medicine over time?
Well, let me just start by saying I'm not a clinician, and a biologist, but from a biologist perspective, better medicine will come from better biology. And we just talk about diseases are highly heterogeneous, and every difference has a different underlying biology. By knowing all of those mechanisms, it will help us get better treatments in terms of precision medicine. So just, an example, right? Diabetes, type 1, type 2, and even within type 2 diabetes, there's so many different types. And the better we understand all the underlying biology, the better we can tailor the treatments.
Yeah, and I think in the future beyond medicine, there's also a rather large field focused on AI and health. And there are a lot of health records, a lot of information about human biology there. But don't you want the health AI to actually understand the foundational, let's say, about cell division, cell biology, to be able to hopefully integrating these information together and give better advice?
Definitely. Do you think there are specific diseases of the brain, for example, neurodegenerative conditions like Alzheimer's, where this direction could be especially important?
Yeah, definitely. Genetics is a huge component to neurodegenerative diseases. And we just talked about the green 2A and 2B. They're playing an important role in intellectual disability. But the same thing applies to Alzheimer's. And it's such a devastating disease, but it's only recently we start to appreciate microglia, this immune cell type, its role in Alzheimer's, right? And it just underscores how important it is to look at cell types and combining that with the genetics and how they manifest in diseases.
Yeah, and I would say as powerful as genetics and human genetic can be, oftentimes what they tell you is sort of the focal point to look at. And the genetics, the causal gene themselves often are not really the eventual sort of target for the therapeutic. We see that in oncology and cancer biology all the time. So genetics is a very effective and strong sledgehammer. But to design medicine out of that, I think there are a lot of examples that people often using functional map to not maybe looking at the primary hitter, but the secondaries. Really is a balance between the effect size and toxicity or tolerability. And I think this is where functional data are helpful. Functional data from the right cell type in the right native context from the disease environment, that's very helpful. But AI integrate this large amount of sort of realistic physiology data can really bring up effective treatments that ultimately help us to understand the biology underlying.
Got it. What do you think it means to move from mapping biology to predicting biological outcomes? So, for example, kind of infrastructure, thinking about data sets and computes to different types of collaboration are needed to make this reliable.
Yeah, we just talk about the confluences of data, compute, and AI models, and just to double click on the AI models, right? In order for it to move truly to a predictive science, that we really need to come up with also better evaluations, and AI models probably not only general models, but also in different verticals. So then, because those comes with the different validation data sets and evaluation, right? And then on top of that, and something that we often ignored, but is critically important, is the people participating in this whole loop. That we want people coming from different perspectives to bring in different expertise, and with the more of those we have collaborations we have, it will help bring all those three ingredients together.
That's right.
So, speaking of people, do you think this changes who can participate in science?
Yeah, I think technologies, and especially in this case AI, what they're really good at is actually to break down the barrier for more people to participate science and enjoying science. And I think you and I probably all experience the time that sort of we need to do a lot of the mechanistic steps to install packages and troubleshooting before we can ask that burning biological question we all want to ask. And I think this is the example that AI can take care of a large portion of that and help us to focus on these questions actually sooner and better. And because of that, I think science will be more interdisciplinary than ever. That really allow us biologists, including molecular biologists, neuroscientists, genomicists, to have a conversation with, let's say, clinicians, because AI can help us to translate each other's languages. And I think that's a really exciting time for more to participate, for everyone to work on these rather ambitious, large projects together.
I would love the day when I no longer have to install any of my own packages and AI can just do it for me. So AI can help us in a lot of different ways. What do you think still requires either human judgment or domain expertise or experimental validation in this world?
Yeah, well, biology we just talked about is complex, it's heterogeneous. So, in addition to, we can get all the help we can from AI, but fundamentally we have to trust and verify, double checking, and exercise our critical judgment, and also designing experiments and thinking about interpreting the output and coming up with a better validations, and all of those are still require and human involvement.
Yeah, I think that also is sort of consistent with how traditionally biologists are doing sort of research in the lab. We all know that any tools, no matter sequencing or imaging or other types of readout, they're not perfect. But usually sort of the weaknesses of any technology, they are not overlapping. And therefore, if you see certain phenomenon using one technology, but you validated this and the intersection is likely to pointing to something that's conserved.
Ladies, I'm just going to jump in because I'm worried that maybe Joy's Wi-Fi is having challenges. Sorry, Joy.
Hi, I am so sorry. I think I have one of those critical Chrome update things that just decided to cancel.
Oh no! Yes.
So of course, in the 24 hours of the day, I will do it right in the middle of a live stream. But I am back.
That has happened to me before, Joy. No problem at all. Thank you so much for being here. You shouldn't even be working right now. We know you were not feeling well, so you didn't want to come into the office. No big deal. And we totally apologize to those in the audience for the technical challenges. We decided to make this work today because we really wanted to host Perturb AI. We made it work. We're all in community here. It's okay. Things don't have to be perfect. They just have to be good. And this is really amazing, awesome research, ladies. So maybe we could just jump into some audience Q&A because we have so much. I don't even think we could get through them all, but this is so fascinating. And I also wanted to share with you ladies that we have really amazing community members present right now. Dr. Shahram Yazdani, he's the faculty chair and faculty and chair of UCLA Pediatrics. We have Sarah Banya, assistant professor of the Chapman University. Just so many cool people here to listen to your research and your talk, Joy and Grace and Shin. So I'm really glad we made it work. It truly is okay that it's not perfect. We are human. And Joy, we also hear your babies in the background. How many babies do you have?
It's just one, but she is very loud.
How old is
she is 1.5.
Awesome. Those are the good days. Okay. Let's jump right into our questions. So this is from Sarah Banya, assistant professor at Chapman University. How can you casually target or causally target genes? Maybe my definition of causal is from economics, but wondering what that means more specifically.
Yeah, so we leverage the wonderful advances in gene editing. In this case, we use CRISPR guides with AAV advanced delivery, proprietary delivery technologies to get it to a specific parts of our body and tissues.
Awesome. Okay. And Jason DeLuca asks, what new AI techniques are making it possible to model brain activity as a live system in near real time, and which seem most promising for future cognitive enhancement therapies?
I think this is eventually a question for Joy. But I think, for the brain, it is one of the most exciting organs. I would say I'm obviously biased, but I think the complexity really coming from the fact that, there are many brain cells and they're intelligently wired together. They are constantly, sort of communicating with each other as you're typing the question, as I'm answering questions. So it boils down, if we're trying to understand the brain as a data science, then what kind of data is sufficient and at what resolution do we want it? But I'm sure Joy has more thoughts as to that. I think it's a really open and exciting question.
Yeah, definitely. I mean, I think thinking about actually modeling the brain is probably like coming from a reasoning model perspective. I would think of our models more as kind of like a really good ML engineer to actually then build a model of the brain, as opposed to like kind of doing it directly inside of like a GPT model. But yeah, I think this is a field in which we're seeing kind of a lot of uplift from the reasoning models right now. I think we're seeing kind of like superhuman performance already in this field. So yeah, really excited to see where it goes.
Awesome. I also just want to take a moment to let you ladies know that we have many members, research scientists from the national labs like Los Alamos here too. So that's super rad. Okay, diving back into the questions. This is from Adam Zimmerman, Associate Director of Analysis and Information Management at Florida International University. He asks, what's the biggest scientific bottleneck before predictive biology can reliably guide real longevity interventions using blood panels and longitudinal aging biomarkers?
Yeah, I think aging is one of the most fascinating question. It is a phenomenon that's conserved, highly conserved in almost all species on this planet. I would say, sort of going back to this, like, descriptive biology versus prediction biology, right? A lot of things are associated with aging. When I age, my hair got whitened. But whitening, sort of dyeing my hair, is not really rejuvenating my hair follicle, right? So to some degree, these are age-associated changes. And it's much harder to thinking about, when you have such a long time for the cascades of changes to happen, who are the first responder, who are the secondary, who are sort of compensatory, which obviously changing these downstream factor may not help with age itself. And I think from sort of the biological understanding, we know so much about aging clock, the chromatin changes, cellular dynamics. But I think the question here is, when a cell age or on an organism age, many things happen. So amongst all the things that happen, how do you draw a causal inference in that to know that across, let's say, thousands of genes that change, which is the one that we should reverse that's sufficient to change the cellular age per se? And then there are some really, really exciting example out there, such as the Yamanaka factor. This is Nobel-winning sort of cocktails that can bring any terminal differentiated cells back to this ground state stem cell. So that's an extreme example. You probably don't want to do that too often. But I think the question is, can we use AI to understand what drives age, first of all, what drives age differently in the skin and the brain and other organs, and ultimately to design that as therapeutic? I think that's fascinating.
Yeah, and this is why we're such a fan of causal in vivo biology, right?
Right.
So then we can now take a systems of view, making all those changes and measuring their impact, and then use AI to help us really understand and then effect change.
Awesome. Shin, I also want to raise your awareness that your colleagues are here from Scripps University. I see Marco Uitiebo in the audience. They're here to support you. Yay, awesome. This is truly a community. So just since we can't see their faces, I want them to know that we know that they are here. Okay, awesome. This is from Saman Hussain, a scientist at NIH. Do you think such a CRISPR atlas could be built with single cell imaging data of the brain as opposed to transcriptomics? Would GPT be as useful for image analysis?
Yeah, that's a great question. Can we actually start building causal perturbation atlases across the whole brain and ask how the neuron firing and their patterns of activity changes? And I think that's definitely one of the ongoing directions that we're working on. But I'm super curious to hear from Joy, if we have this data, let's say tomorrow, how do we analyze these sort of like each perturbation longitudinal data and make sense out of them?
See, Joy,
Yeah.
I've done without you here today.
Yeah, this is a good question. I feel like the model's current capabilities I think if you just gave it a bunch of images of cells, it's probably not very good at doing that zero shot. This is something we're looking at improving right now, or in the near future, kind of like for the model to really start to understand these. But I do think the model would be able to write some really good scripts for analysis. And so I think you can actually get pretty far where even if the model kind of like looking at the images natively can perceive them very well, I think it will be able to still do a lot of really good analysis. So I think like kind of thinking of this as a like really smart human, like a postdoc in the lab or something analyzing the data, they probably wouldn't be looking at all the different images individually. They would also probably be scripting. And so I think the model will already be able to do this quite well.
And I would just say, if my experience in ChatGPT with single cell, it's an example, I can only imagine what ChatGPT will do in a few months or in a short period of time. And I continue to be amazed by its capabilities.
Joy, maybe you can take the first stab at this one. This comes from Oliver Pere, quant researcher. Do you think general large language model based AI like GPT Rosalind will have more impact on biology than modality specific foundation models?
Yeah, that's interesting. I think they do very different things. The example that I gave a little bit earlier is kind of like thinking about what Rosalind does versus a foundation model. I think right now the way that they interact is mostly kind of like in terms of tool use. So I think a common example people use is kind of like you think of AlphaFold and how to hook that up to a large language model. I think using a tool like AlphaFold or Bolt, etc., correctly and kind of understanding the outputs and trying to see if you get an output out, is that still like a high quality prediction? All of this actually requires a lot of expert knowledge. And I think that is where the large language model can come in. So I think you really need both to kind of push the frontiers of science.
Awesome. Thank you, Joy. So Peter Bryant, adjunct professor at IE University, asks, to what degree can your method expose intracellular mechanisms? Or will the method point to the intracellular effects for a specific cell type that require further investigation?
Wow, neuroscience aficionado. I love it. So I think the video that Grace showed is a depiction of how this is entirety of a full brain. And what we're seeing is individual labeled neurons. You all see that they have different beautiful cytoarchitecture, connect to different parts of the brain. So each individual neuron that you see that's labeled actually harbor a different mutation. So that's exactly the type of data that we're also collecting, not only looking at when a mutation happened to me, I'm a cell autonomously, intrinsically what happened, but also how does it change my ability to talk to the rest of the brain? Because I think ultimately the brain is intelligent because it's a bunch of cells that intelligently wire together. So to study how they interact with each other and how their effect changes when the environment changes is definitely something that we're very excited in sort of generating this kind of imaging data, capture these complex interaction and further analysis them.
Okay, maybe two more questions, ladies.
Great.
Let's take a question from a non-technical research scientist in the audience. We have Nicole, VP of Marketing for S2 Genomics. She says, these are incredible results. I understand that you can quickly cross-check the metrics. Hallucinations come with big consequences when applying AI to biology and medicine. How do we mitigate these risks?
Yeah, I think hallucination is still kind of a frontier problem that is difficult to fully solve. But I think from GPT-3 to now, we've made a ton of progress. I think especially for this use case, everything that the model concludes is very well grounded in different citations. So here the model is pulling actual analysis and patterns out of the data. And when it does that, it's associated with the actual analysis that it's done. It's cross-checking different databases and publications. And as it does that, it also kind of cites where these different types of information came from. And so I think like by forcing the model to be very grounded in this case, we then also have confidence that there is no hallucination in these conclusions. But of course, it's also for something that's like very high consequence, you want multiple layers of kind of mitigations on top of that. And so for here too, we also kind of have the original agent does the analysis, and then we also kind of have like a judging model, then go back over and check its work and so forth.
Yeah, one thing that brings my mind is that, this famous story about talking dog. And Natalie and Joy, have you heard of this? It's it's by this famous theoretical neuroscientist, Terry. And what he said when ChatGPT came out, it was that there's this talking dog, and people are just so amazed about it that they doesn't really even matter what the dog is saying. And I think this applies to this question that just because that the language models and AI have been giving us something, it doesn't mean we need to suspend our judgment, right? We need to be critical and think about and bring orthogonal evidence together and say, does it make sense? In what are the uncertainties that we should be careful about?
I love that analogy. Okay, last question from the audience, ladies. This is from Medina Akan, PhD researcher at HGU. How can your AI model the cascading ripple effects of a gene modification across a network rather than just target one to predict unintended impacts on overlapping genetic pathways?
Yeah, this is one of the wonderful things that came out of our data. I don't know if you remember this heat map we showed with green 2A and B, that the heat map kind of it's essentially a representation of the network, and it emerges from our data rather than us specifically looking for it. What it nicely does is to group all the genes that are in a similar network together, and those are colored red. We didn't go into those details earlier, but then there are also the ones that are in opposite networks with opposing effects. They're colored a different color. So it's a really nice way of knowing not just how the genes are playing an important role in an individual cell, but how they're communicating with each other and how cells are communicating with each other. And so what I love about, one of the core things I love about this kind of a data set.
And this data set is released, so is our manuscript. So check it out from our website.
Definitely. And maybe we can drop that in the chat. Well, ladies, that brings our talk to a conclusion. It was wonderful spending time with you in the OpenAI Forum. And Shin and Grace, you're in the San Francisco office at OpenAI. So I think you have some really fun lunch dates after this. So glad you were able to join us in the office. Joy, thank you so much for showing up today. I do not think I could have hosted this without your expertise. I hope you take really good care today. Spend the rest of your day resting. We're going to completely not bug you for the rest of the day. I promise. Space. But it was really, really a pleasure to host you, ladies. Thank you so much for being here in the OpenAI Forum. Grace and Shin, I hope to see you soon for another in-person event, a workshop that you're going to be contributing to. And Joy, I just cannot wait to see the incredible work that comes out of your team in the future. We will be in close connection, and I hope this is just the very first time the forum gets to host you. For all of you in the audience, thank you so much for being here with us today. This is our last event in April, although who knows, crazy things spring up all the time. Maybe something else will land on our plate tomorrow morning. But we will be publishing all of May's events very soon. So keep an eye in your inbox for the OpenAI Forum newsletter. That's where we post all of those events. And we're going to have a sneak peek of we have 2 in-person events in May, this coming up month, and one of them is going to be in DC. So if you're in the DC area, especially, please keep an eye on your inbox. Shin, Grace, Joy, have a wonderful rest of your day. Thank you for being here. And to the community members, we'll see you soon. Marco, it was really cool to see you again. Good to see you. Bye, everyone.
Thank you, everyone.
Thank you so much, Adelaide.